Hi, I'm Philip Tran, and today I will be discussing the research that my gemstone team is doing. We're called Team Vessel, and that stands for Vascular Electrospun Silk Engineered Linings. As you can see from the graphic on the left, cardiovascular disease is the leading cause of death around the world. Our team is focused on solving the problem of peripheral arterial disease, which occurs when there's plaque buildup along the walls of blood vessels, restricting blood flow. A solution to this problem is the use of a bypass graft, which, as you can see in the graphic on the right, circumvents the plaque-filled blood vessel and allows for regular blood flow. This is typically done using an autologous graft, which is made by harvesting the tissue from the patient, but this can be complicated due to lack of tissue or poor tissue quality. So what we're seeking to do is to create a synthetic graft using silkworm silk that will be able to provide the same properties, both biocompatibility and mechanical properties, of an autologous graft. Here you can see the timeline for our methodology. Currently, we're somewhere between the third and fourth steps of our project. We've already completed the electrospinning, and we understand how to fully do that, but now we're testing different growth factors as well as the mechanical properties of our grafts to collect data for our upcoming thesis conference. The picture on the left is a graft from a research paper that's similar to what we're trying to produce. We're creating ours from silkworm silk, as you can see in the right-hand picture. Those are our very own silkworm cocoons donated kindly from the Tufts University lab that's also conducting similar research to ours. However, we're conducting a unique experiment in that we're adding different growth factors and materials to our silk to improve the qualities of the graft. My contribution to this project has been the electrospinning of the grafts. Electrospinning works by creating a voltage difference between the tip of the needle and whatever material you're electrospinning onto. As you can see from the graphic on the upper left-hand corner, this allows the silk to leave the syringe and go onto the rod or whatever surface you happen to be spinning on. So the bottom left-hand picture and the right-hand picture are images of the grafts that we make and part of the setup of our team. We are trying to create small diameter grafts, which is why the graft on the left-hand side seems so small. It's also shrunken a little bit because it's dried, and when it's wet it will grow in size. We handmade our entire electrospinning setup, as you can probably tell from the wood blocks and the electrical tape everywhere, and we do not have a very professional setup. However, we are able to produce grafts that give us really good results and mimic those found in literature. Currently, we only have preliminary mechanical testing data. The figure on the upper left-hand side shows a stress-strain curve, and our graft failed at about 17% strain, which is comparable to those found in literature. However, that was with testing from a sheet of electrospun material instead of a tubular graft, so we've yet to conduct tests with that. The bottom left-hand image is what we use to conduct tensile testing. So what happens is the graft is placed inside the ring and clamped on both ends, and then the ring is slowly twisted to create torsion, and that allows us to test the durability and mechanical strength of our graft. The image on the right-hand side is what we use to test burst pressure. So that's what we're going to use to determine if our graft is able to handle the blood flow found in humans. And this is done by inserting a catheter into the tube and filling it with air and seeing at what air pressure the graft will spring a leak. Our research is attempting to fulfill the need for a viable small diameter synthetic graft. Currently, we're conducting tests using matrigel and endothelial cells to coat the outside of the graft to help improve the performance in the human body. We hope to finish conducting our biocompatibility and mechanical testing in the next couple of months so that we can wrap up our research and present at our thesis conference. I hope that our project is able to inspire further research in this area and that we are able to produce results that can positively impact the field of cardiovascular disease and health.